In this video, we will discuss about how this intracellular detection of self and non-self epitope by the innate immune response occurs. In this video we will consider three different cases. First one is missing self, aberrant self, and the non-self. So we will start with the missing self. So whenever self epitopes are formed using the membrane, using the host cell membrane, it contains this IgRM that is shown by this green color, and this IgRM will prevent this GKS activation. That is, GKS is usually resides in the cytosols of the cells. So in order to get activated, the GKS, it needs to convert this GDP into the GTP, and this conversion is inhibited by this IgRM, which is present within the self epitopes, or the vacuole that contains the self epitopes. So GKS will not be able to bind to the IgRM guarded self vacuole, whereas pathogen containing vacuole is devoid of this IgRM, and because of this GKS activation occurs by converting GDP to GTP, and thereby this GKS can be easily binds to the pathogen containing vacuole, and this after it's bind to the pathogen containing vacuole, it will disrupt the vacuole of the pathogen containing vacuole, and thereby this pathogen uh, get exposed to this galactin 8 that helps in that with the help of this NDB52 helps in xenophagy. And xenophagy, in short term, we can consider it as autophagy, to take care of the foreign particles within the cells. So, and in the second part, that is aberrant cell, what happens is that whenever a vacuole is formed, it is decorated by the glycans on the plasma membrane, and also within the lumen of the vacuole. And this thing happens both for the pathogen vacuole and also for the self epitope containing vacuole. And in pathogen containing vacuole, this glycans disrupts the integrity of the membrane, and because of that, it leads to the exposure of pathogen to the cytosolic containing galactin 8, and the cytosolic containing galactin 8 can interact with the pathogen and also recruit this NDP52 that I have just said for the missing cell part, where the GKS disrupts this vacuole, and then pathogen is exposed to this galactin 8, and these interactions, when the galactin 8 interact with this pathogen, it recruits this NDP52, and that helps in this xenophagy, and thereby the pathogens that are able to enter the host cells will be degraded either by lysosomes or by autophagy. And last one is this non-self epitope. In this case, when the pathogen directly enters within the host cells, this pathogen will be detected by these receptors. The pathogen recognition receptors will be detected by this LRSAM1. These are present within the cytosols of the host cells, and as soon as it interacts with this uh, pathogens recognition receptors, it recruits this NDB52, and simultaneously the polyubiquitations occurs at that site, and mainly this K6 and K27 polyubiquitation occurs, and this NDP52 recruitment helps in this xenophagy, and this polyubiquitation of this K6 and K27 type helps in this also helps in this degradation of this foreign bacteria that are able to enter the host cells. So although the three different terms are shown here as independent approaches, however these three approaches uh, can occur simultaneously, and after one step, the second step can take over and helps in this degradation of the foreign antibodies, degradation of the foreign pathogens that are able to enter the host cells, and all this process is done by the innate immune response. So see that these are the innate immune responses, as these are the first line of defense, and also these uh, this does not generate any specific uh, antibodies against the foreign detected particles. It has already has the information about pathogen recognition particles or the non-self epitopes, or in other terms we can say that it has the information about the self epitopes. So whenever they find something that does not have the self epitopes, it generates a innate immune response and degrade those pathogens via lysosomes or via autophagy. So I hope you got an idea how this innate immune response works within the intracellular of the host cells. If you need to understand more about this intracellular innate immune response, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.